Video instructions for your lab for tomorrow. I want all the groups to be able to complete your cell lab, in which case this cell is going to be involved in the egg experiment for the cell membrane. One person in your group is going to be using gloves, but only one person in your group only needs to put these gloves on. What you want to do is get your egg. You're going to notice the egg still has vinegar in the bottom. You're going to notice your egg is very crusty. Simply go over to a sink, dump all the vinegar out, keep the egg inside, the way I see it here, and that way you're able to get all the vinegar out. You don't need it anymore. Second, your hand with the glove on. I know my hand doesn't have a glove. Your hand with the glove on, you're going to roll this onto your hand. That egg is then going to be put into this plastic cup because your first objective is to find the mass of the egg. I don't want the mass of the egg in the cup, I want just the mass of the egg, so it's your job to determine how to do that. You're going to notice there's 3 triple beam balances. That means there's also 3 cups. Please share. Once you find the mass of the egg, and you're going to do it for both your eggs, you're going to put this egg, in this case, this one says water, you're going to put this egg in water. Fill the water cup to where the egg is completely submerged. Nothing more, nothing less, just right to where it covers the entire surface of the egg. Do the same one for corn syrup when it asks for corn syrup. You're going to have a time interval of how much time you want to wait as the fluids start to interact with the cell membrane. You can wait 3 minutes, 5 minutes, that's up to you, that's to be determined. That's going to be put in your scientific report. Once you determine that time, let's say 3 minutes, time's up, take the egg out, put the egg in a plastic cup, follow the same procedure on how to measure the mass of just the egg. Once you have finished the mass of just the egg, put the egg back into the same container, take a time again. While that's happening, you can do the same thing with the other egg, get it done, and every time you have to put an egg in here, quickly rinse. Make sure there's no more corn syrup inside, make sure there's no more water, no more vinegar, rinse, and so for the other girls to use. You're gonna notice your egg is done because your egg is gonna, 1, plateau, where it's gonna form its own isotonic solution, or 2, it's gonna burst. If the egg bursts, please throw it away. Throw the egg outside in the trash cans outside of the school. Recycle these, do not throw these cups away. Once you're done with this, you may toss this again in the trash can. After you have done your goal, you want to complete a scientific report by having a problem, hypothesis, procedure, which lists materials and steps, results and conclusion. Your conclusion telling me if it's an isotonic solution, a hypertonic solution, or a hypotonic solution, which should be determined based on the results, your masses of the different scales that you have. Hopefully you're able to get at least 5 masses, and again, every time you take the egg out, measure the mass, just put the egg back in. You don't have to refill the container anymore. It's already doing its job. If you have any questions, please email me. Other than that, I wish you the best of luck, and this should be completed by the time you exit the class for today. Everything else, you can put that off to the side, and we'll get to it later as we start our next project and as we start our final touches of the medical journal.